Oh, no, okay. Hi, good morning. So I'm Gabriela, and my topic is the effect of Okadaic Acid, DSP, and its influence on a host microbe interactions in a GNOTOBIOTIC Artemia system. I did this work with my promoter and supervisor, Peter Bossier, in Ghent University. So first, I'm going to give you an introduction about the status of aquaculture, about DSP, Okadaic Acid, Vibrio, harveyi, Artemia, and how all of this combines to form my objectives. So first, about aquaculture. Aquaculture nowadays represents almost 50% of the whole food production. So for that reason, it's very important to try to investigate any diseases or any threats that may occur in this sector. So one of the threats that is currently happening nowadays is the DSP, that is diarrhetic shellfish poisoning, and it's caused by two genus of dinoflagellates, Dinophysis and Protocentrum, and they produce the marine toxin Okadaic Acid. These toxins are produced in harmful algal blooms when harmful algal blooms happen, and they can accumulate through the trophic web, so they can accumulate in the primary producers to primary consumers and secondary consumers, and then when humans consume these organisms, especially shellfish, it can reach to humans. So the mainly symptoms that occurs are nausea, diarrhea, and abdominal pain. Another threat that happens also can reach humans, but happens especially in aquaculture, is vibriosis caused by several types of Vibrio. In my study, I will use Vibrio harveyi. It's a luminescent bacteria. It has worldwide distribution, so it can be found anywhere. And Vibrio is the main causes of vibriosis. As you can see in this photo, vibriosis especially can happen in shrimps and mollusks. And in shrimps, it can cause lesions. This is a healthy shrimp, and these are two sick shrimps. And they can cause lesions in their bodies and also make their color change. So Vibrio harveyi can do quorum sensing. Quorum sensing is a cell-to-cell bacteria communication where they can control their virulence using autoinducers. So in my study, there are three main autoinducers, and in my study I'm going to use 5 strains of Vibrio harveyi. So about the category of autoinducers, this strain has all of these 3 autoinducers. This next one has only the last 2 autoinducers. These, the first and the third, and these, the first 2. And for the last strain, it has no autoinducers. So these are the strains that will be used. And Artemia is known as brine shrimp. They are considered model organisms, so that's why they were chosen for this study. They are found in inland salt lakes and coastal saltwater's, and this is their life cycle. They can reach adulthood around 5 to 6 weeks, and this is the stage that I use, that is the Naupli stage. So, with these harmful algal blooms and all the marine toxins they can produce, we want to establish if the Okadaic acid is an important parameter in host microbe interactions, and also if it can facilitate a Vibrio infection in Artemia. So for that, I had 4 tasks. The first task is the standard method to do this is done in sterile glass. And since Okadaic acid is a very expensive product, we're trying to do it in a smaller scale experiment. So I use 96 microL plates. So the first task is to adapt this methodology to a smaller scale experiment. And the second one is to test the toxicity of Okadaic acid in Artemia, because this was never done before. And I performed 2 challenge tests. One, testing if Okadaic acid established an interactive effect when combined with Vibrio harveyi, and one, testing different strains of Vibrio harveyi, and if it combined with Okadaic acid, can establish shifts in virulence. So my material and methods. So first, the culture of Artemia. Artemia was obtained by INVE Aquaculture Company in Belgium. They arrive like this in the forms of cysts. And first, these are the cysts seen without hydration. These are the cysts with hydration, and these are the cysts after decapsulation. So first, we do decapsulation to make them sterile, and then we leave them for hatching for 24 hours at 28 degrees. About the feed preparation, the Artemia, they were fed with LVS3 food source. They are first spread in a plate, and then they are grown, and after that we choose one colony and we transfer to marine broth. We autoclave it, and then we measured the final density to have a final concentration of 10 to 7 cells per milliliter. The pathogens, the 5 strains of Vibrio harveyi, they were spread in 5 plates also. And after 24 hours, we also chose a colony, and we also transferred to marine broth. And we also used the final concentration of 10 to 7 cells per milliliter in the experiments. The Okadaic acid was obtained by Abcon, and it was dissolved in DMSO, and it was kept in minus 20 degrees Celsius. So the experiment. The first experiment aimed to understand the toxicity of Okadaic acid towards Artemia, and if there is an interaction with one strain of Vibrio harveyi, that is BB120. So this experiment, we used 10 naupli per well. We did 5 replicates, and we used a total volume of 100 microliters. So in this side, you can see that we had the control and 6 different concentrations of Okadaic acid, and they were chosen to cover, according to previous studies, they were chosen to cover all the possible lethal and sublethal concentrations. And this side is without the strain BB120, and this side is with BB120. So the next experiment aimed to understand, test different strains of Vibrio harveyi and see if when they had Okadaic acid, if they can stop shifts in virulence. So we also used 10 nauplis per well, 5 replicates, and 100 microliters in total volume. But in this case, we tested this. You can see that there is a control and the 5 different strains of bacteria that we used. And this side, we had absence of Okadaic acid, and this side, presence of Okadaic acid. But we tested also 2 different concentrations of Okadaic acid. So in this case, we used 10 nanomolar, and in this case, we used 20 nanomolar of Okadaic acid. And we count survival for this and the previous experiment. We count survival after 24 and 48 hours. So about the results. So first, analyzing the effect of the different strains of Vibrio harveyi on Artemia, non-significant difference was found in any of the strains of Artemia. This is the average survival rate of the Artemia in percentage calculated, and these are the strains that we used. So for 24 hours and 48 hours, we found no significant difference. And considering the effect of only Okadaic acid on Artemia, also having the average survival rate over the different concentrations, we found that for 50 and 100 nanomolar, a significant effect that shows that they have high mortality in these 2 concentrations, both for 24 and for 48 hours. Now analyzing the interaction effects. The interaction effect between Okadaic acid and Vibrio harveyi. So the green bars represent absence of Vibrio harveyi BB120 strain, and the blue ones represent the presence, and this is the different concentrations of the Okadaic acid used. So these results are after 48 hours, because the results after 24 hours had no significant interactive effect. So these results show that we can find an interactive effect in 2 concentrations, 10 and 20 nanomolar. 10 and 20 nanomolar. And analyzing the interactive effect between Vibrio harveyi, between the 5 strains of Vibrio harveyi and Okadaic acid, First, when we had the presence of okadaic acid at 10 nanomolar after 24 and 48 hours, the green bars representing absence and the blue presence of okadaic acid, and here representing the different strains, we found an interactive effect in only one strain, but we couldn't find the same effect after 48 hours. And when we analyzed the presence of okadaic acid for 20 nanomolar, we could find for both 24 and 48 hours an interactive effect in the same strain, that is the strain BB120, the wild type. So about the discussion, okadaic acid is toxic to Artemia for over 20 nanomolar concentrations, as we could confirm from previous studies. And when it's combined to the pathogen, they are toxic at 20 nanomolar, and they have an interactive effect. BB120 strain, that is the wild type strain, they have an opportunistic behavior, because when it was alone, it didn't cause mortality, but when it was combined with okadaic acid, it caused mortality, and we found no differences in quorum sensing. So, to conclude, Artemia were exposed to Artemia, when they were exposed to 50 and 100 nanomolar, they were lethal. When they were exposed to 10 and 20, they were sublethal. But when we combined 10 and 20 plus the presence of Brine, they became lethal. When they were exposed to BB120, they were not lethal. But when they were exposed to BB120 plus this 20 nanomolar of okadaic acid, they became lethal. And comparing the controls, the survival results with previous studies, the small-scale protocol was considered very efficient. So for recommendations for future researchers, the first one is important to investigate the toxicity of okadaic acid in another organisms, because we have information about mice and rats, but I think it would be important to investigate, especially in shellfish, because it's the main pathway that they can reach humans, and also to use this test also to confirm the results. Also, it's important to test not only seasonal variations, but try to have an annual variation of this concentration during the year. And because we only have information of okadaic acid in seawater during some months per year, so I think it would be important to see how it varies during the whole year. And see if there are months that they are higher, or if there are months that they are lower because of the harmful algal blooms. And also, I think it's very important to research other biotoxins that are resulted from harmful algal blooms. So, that's it.